Hey everybody, we are going to continue on talking about DNA and replication, and we are going to take a look at what happens after elongation is over and the DNA molecule has to be proofread. Do errors happen when DNA replicates? Yes, but they're very rare. So about 1 in 1 billion base pairs, there's an error that kind of averages out to about 3 mistakes in our DNA each time it's replicated. So when you're talking 3 mistakes out of a few billion base pairs, it's a pretty good error rate. But there's reasons for that. So why is it that DNA replication is so accurate? Oh, you have the DNA polymerase III that's going to self-check and then correct any mismatches. And then you have a whole nother family of enzymes, and their entire job is to check and then correct any errors that they find in DNA. Right now, there's over 130 different DNA repair enzymes that we know about. And so they're pretty good at their job because failure to repair any of these errors could lead to cancer or other major health problems. Just a really quick example is xeroderma pigmentosum. This is going to be a genetic condition where a DNA repair enzyme doesn't work. You're going to have UV light cause damage, and then that damage is not going to be fixed, and so it can lead to cancer. So here you see like they look like moles right here. So that is skin cancer. So that boy is having some issues. You don't expect to see skin cancer like that in a child. So one type of error caused by UV light that we're going to look at right now is thymine dimers. And so these are when you have thymine bound to each other side to side. So instead of the thymine binding with the adenine across from it, it's going to bind to another thymine right next to it. And so it causes a backbone in the DNA, it causes a bubble in the DNA backbone. And so normally this is repaired. You just cut out that damaged section of DNA. DNA polymerase will come in and fill in that area with new bases, and then your DNA ligase will seal the backbone up. So here is the example. So these are two thymine, and again, instead of hydrogen bonding to the adenines across from them, they are binding to each other. So you'll have an enzyme come in, cut out that section with the error. DNA polymerase will come in and add new nucleotide bases, and then DNA ligase will seal it all back together. So we have another problem that we need to talk about, and that is the ends of DNA molecules. So we said yesterday that DNA polymerase can only add nucleotides in that 5' to 3' direction. And so what that means is it's going to end up running into the problem of not being able to complete the ends of the DNA strand. So with each round of replication, that DNA is going to get shorter and shorter. So this is kind of an example. Here you have your parental strand of DNA, and here is your lagging strand that is being made. So the red areas are going to be your RNA primers. So this primer was laid down, and then the DNA nucleotides were added. Then this primer was laid down, and then these nucleotide bases were added as well. Now we know that you're also going to have to remove those RNA primers and replace them with DNA nucleotide bases. And so in this strand you can see that that was done, except right here. We're unable to add in the nucleotide bases because this is the end of the DNA strand. So there's nothing for that enzyme to grab onto and go in and add those new bases. So what happens is this DNA molecule is now shorter than the parent molecule. And so now after round after round after round of this DNA molecule will get shorter and shorter. Now there's a way that this is dealt with, and it's called telomeres. These are going to be repeating units of TTAGGG. And when we say repeating units, we're talking like 1 to 1,000 of these at the end of a DNA strand or at the end of a chromosome. And it's going to protect that DNA from unwinding and sticking together. And the telomeres are going to be what's shortening at each DNA replication. So you're going to end up cutting off ends of these telomeres as opposed to that DNA that you're trying to replicate. So telomeres can kind of serve as a clock to count how many times DNA is replicated. And when the telomeres are too short, the cell is going to receive a signal. And we talked about this a couple chapters ago, that cells have this programmed cell suicide, apoptosis, and so a cell is going to essentially kill itself. It's going to receive the signal to do so anyway when those telomeres become too short. So telomeres are involved with the aging process, and we have an idea that they limit how many times a cell line can divide, because at some point they're going to get too short for that cell line to continue. We have an enzyme called telomerase, and this is going to be an enzyme that uses RNA to rebuild those telomeres. So there's this idea that telomerase can make cells immortal. It's found in cancer cells, it's found in germ cells, and it's limited where you see cells that have a pretty short lifespan. So an example would be the skin cells. So we know that skin cells reproduce rather quickly. We know that you shed your skin very frequently, so like every time you scratch and you kind of see like flakiness on your hand, those are skin cells that are leaving. So your skin cells have a much shorter lifespan, and so they don't necessarily need to be rebuilding those telomeres. Alright, so here we are looking at our telomere. So we have our repeating units here. So you got our TTA, GGG, TTA, GGG. So those are your telomere, okay? So the telomerase extends the 3' end of the DNA strand. So you can see that's happening right here. We're adding in, so TTA, and it would be the GGG. So the other strand is extended in usual way by primase, DNA polymerase, and ligase. And so now, because we've extended this 3' end, it means we can extend this end as well. All right, and so the result is a longer telomere with a 3' end overhang. So you can see when we talk about the overhang, that is this part right here. So 3' end is a little bit longer than this end down there. So just general thoughts on this. Control of telomerase may stop cancer. It could potentially extend lifespan. So really, there's some interesting implications here. Obviously, we need some more information, but it's kind of interesting to think about that there's an enzyme that can, stop cancer, extend lifespan, essentially by extending the ends of your DNA so it doesn't shorten, or it's still going to shorten, but it doesn't hurt it as much when it is replicating. So here's your lecture question for today. So the DNA of telomeres is actually used to build protein. So the TTA-GGG, that codes for something, okay? So it's used to build proteins, and these proteins seem to actually impede the telomerase. So do you know why that might be? Why would the DNA of the telomeres actually be used to build proteins that would stop telomerase? So see if you guys can shoot me an answer to that question. I'm really interested to see what you think. Thanks, guys.